Hello, everyone. Thank you for joining me today. My name is David Anderson, and I'm a principal scientist on the Advanced Technology and Analytics team at Clinical Inc. And I'm excited to share with you today some of our digital biomarker work that we've been doing in Parkinson's disease, specifically within the WATCH-PD study. And we'll be focusing on some of the feature engineering and machine learning applications that we applied to our sensors and wearables used in that study. So for this presentation, I'll be reviewing the WATCH-PD study, some of the feature engineering work that was done, our machine learning application, and discuss some of the conclusions and applications of this work. So the WATCH-PD study was a collaboration between the University of Rochester, Biogen, Takeda, and the Critical Path Institute, with Ray Dorsey and Jamie Adams from the University of Rochester serving as the principal investigators. The study was designed to develop a core set of digital biomarkers that would map directly onto the MDS UPDRS using a comprehensive battery that assessed multiple domains affected by PD, including cognitive, psychomotor, voice, and mobility tasks used within our Clinical Inc.'s brain baseline movement disorder platform. With these biomarkers in hand, the goal was and continues to be to work through the critical path to have discussions with the FDA to move these towards digital endpoints for approval for use in studies of early stage Parkinson's patients. The platform itself was completed on the phone and the watch that were provisioned to patients upon enrollment into the study. The application was comprised of an ePro that evaluated things such as self-reported tremor severity, thinking, concentration, mood, and medication adherence. Several cognitive assessments were used that evaluated things like processing speed, attention, working memory, and executive function. And we also evaluated fine motor and finger tapping. In addition to having primary endpoints that are standard use in neuropsychological and cognitive science paradigms, which these were modeled after, we also were able to generate millisecond by millisecond sensor data from the phone surface or screen that told us where the kind of movements were on the device to give us more granular insights into user behavior and performance. We also evaluated 3 core speech assessments, specifically looking at verbal phonation using the sustained ah task, verbal articulation using the pa ta ka task, and verbal reading using Harvard sentences that varied across each administration to minimize practice effects. Each assessment was completed on the phone and digitized at 32 kilohertz. We also had patients complete motor assessments, and for these they completed them on both the phone and the watch. The watch was worn on the most affected wrist for the patient group or on the non-dominant hand for the comparison group. The phone was worn on a pouch connected to the hip, so essentially measuring from the trunk. Participants completed a gait task in which they were asked to walk as they normally would for 60 seconds, and a balance task where they were asked to stand as still as they could with feet shoulder width apart for 30 seconds. They also completed 2 tremor tasks, one measuring postural stability and the other measuring resting tremor. And we collected the same data from both devices, both sensors, specifically measuring from the IMU at 100 hertz. So we were looking at acceleration, gyroscope, and magnetometer data. In addition to these active assessments, passive data collection was performed that was measured 12 hours each day, and specifically we measured acceleration and predictions of tremor severity and whether dyskinesia was present or absent during time samples. So the study itself was conducted over a 1-year period. A set of clinic visits were completed at baseline in months 1, 3, 6, 9, and 12, which were supervised by a site coordinator. And then remote visits were completed at home, which were unsupervised and self-guided by the participants. every 2 weeks. So the data were generated on both the iPhone and the watch, which were encrypted while on the devices and then securely transferred to our brain baseline server hidden behind an AWS firewall, where it was also stored in encrypted format. The data were then ingested into our Snowflake data lake solution, which is used for both ad hoc research internally and for providing customer data as well. In our ad hoc research environment, we performed a series of signal processing routines to generate our feature engineering pipeline and then fed that data into our data modeling pipeline, which I'll be reviewing here next. So for feature engineering, we generated over 3,600 features from the assessments that I just described. This is just a basic summary of the assessments that were evaluated, the domains assessed, how many features were generated from each assessment, and then some examples. So for example, in the gait and balance tasks, we generated over 2,100 features. That assessment was developed to measure slowing of gait, dyskinesia, and bradykinesia. And to that end, some of the features that we evaluated were step frequency, synchronization between sensors worn at the trunk and the wrist, and path length of those sensors measured. We used a combination of time and frequency domain signal processing to develop these features, specifically with the focus being to develop behaviorally and disease relevant measures. To that end, we performed linear regression on each feature to determine which ones would actually tell us something about whether participants were in the PD group or in the healthy control group. So this is just a distribution of the F statistics from those linear regression routines, where the red line here corresponds to the threshold for significance. And demonstrating that these are disease and behaviorally relevant, we found that 39% of the features that we developed were indeed selective for PD status. And so with all of this data in mind across these multi-domain set of assessments, we then wanted to develop a multivariate model that best predicted whether patients were in the PD group or in the comparison group. So that brings us to our machine learning application. For this approach, we took our over 3,300 or 33,000 active assessment data and fed them into our feature engineering pipeline. Once we generated all of our features that I just described, we then fed it into the machine learning modeling routine that separated the data into 90% training and 10% test, where no subject was represented in both training and test data, ensuring independence of these data sets. The features were then fed into feature selection and feature reduction routines, where feature selection was implemented using linear regression to identify the 100 most selective features, and feature reduction was implemented using principal component analysis. To determine the extent to which feature processing influenced model performance, we parametrically varied whether feature selection and feature reduction were performed for each model, resulting in 4 possible combinations of feature processing routines for each model. Whatever we did with the training data was also performed on the test data. We then fed our features into our model development routine, where we evaluated 9 different models and obtained the model parameters that were then used to make predictions about our independent test data. We compared those predictions against the actual labels of whether the participant was in the PD or comparison group, and then evaluated the model performance metrics for those predictions. To ensure that the model performance was not influenced simply by how the data were sorted into training test data, we performed Monte Carlo simulation 100 times to randomize the training and test set allocation of those data. And so this allowed us to evaluate the robustness of each model. So the result of this approach is a distribution of model performance metrics, specifically accuracy, sensitivity, and specificity. So when I talk about model performance in the following slides, I'll be specifically talking about the median value for these distributions. So this is the result of our model comparison analysis. On the x-axis are each model that we evaluated with the legend below. So for example, GNB corresponds to Gaussian naive Bayes. Each model was sorted based on accuracy and descending order, and each color value here corresponds to how the features were processed. So for example, in yellow, this corresponds to features undergoing selection routines but not reduction routines. And this black bar here corresponds to models that did not significantly differ from the best model. And so here you can see that the best model was a random forest model that did not implement selection or reduction routines. And when we looked at the model performance metrics, we observed an accuracy of 92.3%, sensitivity of 90%, and specificity of 100%. And so of course, the best model is going to sound great. And so then we wanted to determine the boundary conditions of this model to really try and break it and understand how it performs across different contexts. And so recall in this study, we had both an environmental and a temporal component to data collection. So the first thing we wanted to do was to determine whether this model continued to perform well across different contexts. And so, for example, we wanted to, different environmental contexts. So for here, we wanted to look at whether we could train on clinic data and then test on data collected at home, for example. Second approach we took was to determine whether this model would perform well across temporal contexts. So could we train on baseline data and then test on data collected a year later? All right, so this first figure is showing the results of our cost environment analysis. What I'm showing here on this far left figure is accuracy on the y-axis. On the x-axis is the results from the model that was trained on, what the model was trained on, and then what the model was tested on. So for example, the second column here corresponds to when a model was trained on home data and tested on clinic data. And then the color of the bar corresponds to whether the data was trained and tested on the same subjects in blue or trained and tested on different subjects in orange. With the idea being that in blue, you may want to develop models specifically for your study and evaluate those predictions over time on the same subjects. Whereas in orange, you really want to generalize across different studies. And so we found a couple of patterns here. First, there was significant interaction between training context, test context, and the model test data sets. Specifically, of course, we found that accuracy was lower when the models were tested on independent data sets than when they were on the same subjects, which is to be expected. We also found that specifically if we focused on when we were looking at dependent data sets, so independent subjects, we're looking at different subjects, we found similar levels of classification accuracy, regardless of whether we trained on clinic data or home data. So these data are fairly generalizable across different contexts. And you could, for example, have in the example that really kind of informed this analysis, you could train on data collected from the clinic and then just test on those same subjects or different subjects at home. So looking at the model performance metrics for this particular analysis, we found that we achieved 92% accuracy, 88.9% sensitivity, and 100% specificity. And you'll recall that this is fairly similar to the results from the model comparison analysis with the same random forest model. And so this really speaks to the reliability of these measurements across different contexts. So the data you're collecting in clinic is going to be similar to the data you're collecting at home. Next, we wanted to look again at temporal context. And so here I'm showing heat maps. This is going to be the same kind of presentation just described with the environmental context, where we're looking, in this case, at train day on the y-axis, and these are 90-day bins. So this first bin corresponds to day 0 or baseline to day 45. Second bin is day 45 to 135, etc. And in the x-axis is the test day. So for example, in this cell here, this corresponds to when data were trained on day 180 and then tested on day 270. The two different heat maps are when we trained and tested on the same subjects or on different independent sets of subjects. So in this analysis, we again found a significant interaction between training test time, and model test data sets. Again, we found that classification accuracy was lower when models were tested on independent subjects than they were on the same subjects. And also classification accuracy peaked when tested on similar time bins with a monotonic drop-off in accuracy from that time bin. So for example, when we trained and tested on day 180, we found really good accuracy when we're focusing on the same subjects, which is to be expected. And then we saw a drop-off in accuracy as day bin increased. Importantly, if we were to train just on baseline data and test on data collected a year later, we continue to see accuracy around 80%. And when we look at those distributions, we find that we did achieve accuracy of 80% up to a year later with 80% sensitivity and 77.5% specificity, which is pretty exciting. All right, so our conclusions, just to summarize, the comprehensive mobile movement disorder platform, which we've developed to map directly onto the MDS UPDRS, supported the collection of over 33,000 active assessments during the WATCH-PD study. We generated over 3,600 features using a combination of a combination of time and frequency dependent signal processing routines on our cognitive, psychomotor, voice, and mobility assessments, with 39% of those features being selected for PD status because we were focused on disease and behaviorally relevant features. We also found that a random forest model most accurately predicted PD status, so this achieved an accuracy of 92%, sensitivity of 90%, and specificity of 100%. And we also observed strong predictive accuracy across environmental and temporal contexts, meaning that these models can be used to evaluate performance across at least within a 1-year time period, and used across both home and clinic environments. All right, so this has implications, beyond just this study. For example, we can use this as a screening tool, and this is particularly important because first we know that the diagnostic accuracy from postmortem autopsy suggests that just using the MDS UPDRS alone, accuracy of diagnosis in early stage is between 58 and 85%. So something like this could complement the MDS UPDRS during screening. And it's also been demonstrated that population-based studies found that 12 to 80% of individuals with PD-related motor symptoms had not yet been diagnosed with PD. So essentially, these patients don't know they have PD yet until they get in to see their neurologist. And so a screening tool such as this that could be used could be beneficial for those individuals. And so, of course, from this, it's plausible that we would apply this to applications for patient screening for studies or for clinicians to use, and supports broader access to patients in future BYOD studies if we wanted to be able to screen across the entire country with individuals using their own devices at home. And with that, we will conclude, and thank you for your attention today.